ganglionic neurons and their effector or target organs. So we're looking at parasympathetic neurons. I'll go a little slow here because probably most of your faculty from last semester didn't go into it as much detail. I actually added a couple of extra days, and I already do more days. So I'll take that a little bit more slowly. Parasympathetic neurons, preganglionic, then are going to release, oh, I don't want to do that. Sorry. Just make one large synaptic hole here. What is a neurotransmitter released by preganglionic parasympathetic neurons? Acetylcholine. And the postganglionic neuron is going to have a channel that binds acetylcholine and opens. Here's where I'm not quite sure how much you covered. What happens to this acetylcholine receptor on the postganglionic neuron cell? Doesn't matter whether it's sympathetic or parasympathetic. We're using parasympathetic for our whole system. So it's always acetylcholine here. When that channel opens, that's a channel for what ion? For sodium. Okay. So the receptor is itself the ion channel. And that's probably what you're used to in all most of the examples that were given. So you're like, so what's the big deal? When the receptor itself is the channel, we call that type of receptor an ionotropic receptor, meaning it creates an ion channel. The receptor itself is an ion channel. Is it always excitatory or always inhibitory, or it can be either depending on the cell? It's always, when sodium comes in, what is it bringing with it?
Excitatory.
Positive sodium ions, so it's always excitatory. So far, this has nothing to do with what we're going to talk about. I'm kind of leading you into it. Does it seem familiar? Those of you that didn't have me last semester, did you cover this much of it? Okay. You may have covered all of it, I'm just not sure. Now the postganglionic neuron is going to form a neuromuscular junction or a glandular junction with, let's say, the pacemaker cells of the heart.
Monotropic.
What's the neurotransmitter? If it's parasympathetic,
ACA.
it's acetylcholine. If it was sympathetic, it would be norepinephrine. All right? So now we have another receptor. Do you see a channel there? No. This is a receptor, and it is a ligand receptor, but the receptor itself is not a channel. It is a receptor, period.
Secondary messenger.
This is where we use secondary messengers. when acetylcholine binds to the receptor, it activates a series of proteins attached to each other known as a series of molecules, known as a G protein. After a series of reactions in this particular cell, a potassium channel will open. As we'll see when we talk about the heart, the potassium will leave the cell, not through the receptor, but through behavior caused by acetylcholine binding to that receptor. This takes longer than this does. It might be excitatory. In this case, it's going to decrease the heart's contraction, so it depends on what cell it binds to and what kind of behavior it triggers in the cell. Sometimes, as it is in the stomach or the small intestine, it's going to make muscles contract. In the heart, it makes muscles relax. All right? In the bronchioles, it makes them contract. So it just depends on what's happening inside the cell. This type of receptor is known as a metabotropic. It requires signals and communication carried on by its cascade of events within the cell. When the hormone binds to the receptor doesn't move into the cell. All right? Let's say that I'm walking down the street, 5:30 in the morning, and it's dark, and I smell smoke. And I can see smoke coming from a house, and it's not coming from the chimney. So I run up to the door, and it's locked. I can't go in. I bang on the door, and I bang, and I bang, and I bang. What role am I playing? Where am I in that? I'm the ACH and the neurotransmitter here. If we're talking about an endocrine target organ, we should have this image. I am the hormone. Okay? The door that's blocking me from going into the house would be the receptor. Okay? I never get into the house. Finally, after 20 minutes, a 2-year-old comes to the door. Can't do much about the fire. All right? But I yell through the door, go wake up your mommy and daddy. By the time all 10 kids are now awake, and they're all running through the house and trying to wake up the adults. So they will be the second messenger, which is a variety of structures that move. In this case, it's cyclic AMP. We'll talk about that tomorrow. Or it might be calcium ions, or it might be other molecules which have very scary names. IP3 and DAG to not scare you off from coming back and learning the names tomorrow. So, today, we're just going to focus on what happens right here, okay, with the G protein. And then tomorrow, we'll pick up the 3 different pathways that the G proteins work, and then how the receptor that looks like this can work when there's no G protein whatsoever. Okay. So, we're going to look at activation of the G protein. And then tomorrow we'll do these areas, all right? G protein and cyclic AMP, calcium channel, phospholipase C, and the variations on that. But they all have in common the sequence that I showed you with the neurotransmitter, which is the activation of the G protein. In this case, and I'll turn the lights back on in just a sec, when the receptor is not binding to the hormone, the G protein is interacting with the receptor. Sometimes, as in the little video I'm going to show you, it doesn't react with the receptor until the receptor is activated by the hormone. Okay, but we'll take with this example here first and go through that explanation. G proteins are relatively recent. I think the Nobel Prize was mid to late 80s. I know when I was in graduate school in 83, 8045, G proteins were recognized, but this whole pathway had not been elucidated yet. So obviously, when I was in college, we didn't have to worry about it. Our brains were smaller back then. So this is a hormone receptor. And it is membrane bound. Remember, our plasma membrane is a lipid bilayer, therefore it's an oily sheen. And this membrane receptor is not always in one place. It can move around. Okay? I'll use the illustration. Our G protein has 3 subunits. In the little video I show you, they have the alpha subunit associated with the hormone. So I won't be making big clarifications like this. When the G protein is not active, it is attached to an energy storage molecule, not ATP, but a form of the inactive low energy level of GTP. So this is adenine triphosphate. This is guanine triphosphate, or in this form, guanine diphosphate, with only 2. It's very similar. There's a high energy bond when that 3rd phosphate is on there. So in the next step, the hormone is out of the bloodstream and bumps into and recognizes and has an affinity for the receptor. When that binds to that, it changes the shape of the protein. Physically, there's a physical change in its shape and its chemistry. And in this particular situation, this G protein no longer fits. And it slides away. It's also soluble in the membrane. So it slides away. And it also now is a different shape because it's no longer associated with the receptor. And with that different shape, GDP no longer can maintain a chemical interaction with the alpha subunit. And GTP takes its place. And our alpha subunit separates from the other molecules. It can move around freely within the membrane too. Obviously, if the hormone is not in its receptor, this still won't fit back into it. It has to go back to GDP. Okay? But we now have the active form of the alpha subunit, which is going to, it's not our second messenger. Second messengers are within the cytoplasm. So we're still on the process of activating that. I'm still waking up that little 2-year-old and trying to get him out to come out of bed, or maybe he's out of bed, he just hasn't come to the door yet. So when it activates whatever molecule downstream, then this goes back to GDP, and it can reassociate once the hormone is gone. Again, tomorrow we're going to look at the 3 mechanisms or pathways by which G protein works. I'm not going to go through them now. I'm just going to introduce them to you, so as you read ahead, it makes a little bit more sense. So that all of these steps are the same. In this case, the alpha subunit chemically interacts with an enzyme in the membrane. And this will catalyze changes in ATP, and those changes are the second messenger. It can do this over and over and over and amplify the signal. Okay? And we'll talk about what components. In addition to that, these are my videos that didn't work. The activation activated alpha subunit can directly open a calcium channel. And the calcium ions can be second messengers. Because the calcium ions are moving into the cell. We saw how that worked with muscle, where calcium ions cause the troponin to move. And then in the third mechanism, and this is the complicated one, we end up activating an enzyme that produces a complicated pathway of other second messengers. But they are all activated with that alpha subunit. So I want to pull out, I got this all embedded in my new computer, but of course it's PowerPoint 10, which we don't, 2010, which we don't have on here, so let me just show it to you off of YouTube.
So the alpha subunit, it's not that, right? Like when she's saying that
No.
A, it's not that, right?
No. There's a picture of it in the textbook.
So if you want to find this yourself, it's G protein activation. That's what I want, doesn't pop up, so let's try to.
Guys, your book. Your fertility. Here.
This is all here. And it's attaching to this.
Oh, to the adenosine-glycosides.
Yes.
Okay.
So it takes this and it turns it into this. And then this loops on over here and opens this channel.
No, it goes for a different site.
Page 46.
Okay. See, what do they call it here? So it's a hormone, this is the hormone receptor, the purple thing, right?
Yeah.
So what's this?
Those are the hormones.
Okay. Hormone receptor, activates the protein gene.
When an appropriate protein ligand binds to this,
Sorry.
the receptor undergoes a concentration
But it's in a different
that is
video.
transmitted to its subsolid
It's in stereo.
which now activate a trimeric GTP binding.
That's double learning right there.
All my new
It's in our book on once. This is actually something we did last semester,
I obviously.
but
that's
it's
what
also
they-
in this unit. Here, it's also on 594.
Thanks, Colleen.
Receptors have a large extracellular ligand binding domain. When an appropriate protein ligand binds to this domain, the receptor undergoes a conformational change that is transmitted to its cytosolic regions, which now activate a trimeric GTP binding protein, or G protein for short. As the name implies, a trimeric G protein is composed of 3 protein subunits called alpha, beta, and gamma. Both the alpha and gamma subunits have covalently attached lipid tails that help anchor the G protein in the plasma membrane. In the absence of a signal, the alpha subunit has a GDP band, and the G protein is inactive. In some cases, the inactive G protein is associated with the inactive receptor,
That's what we
while
just.
in other cases, as shown here, it only binds after the receptor is activated. In either case, an activated receptor induces a conformational change in the alpha subunit, causing the GDP to dissociate. GTP, which is abundant in the cytosol, can now readily bind in place of the GDP. GTP binding causes a further conformational change in the G protein, activating both the alpha subunit and beta gamma complex. In some cases, as shown here, the activated alpha subunit dissociates from the activated beta gamma complex, whereas in other cases, the two activated components stay together. In either case, both of the activated components can now regulate the activity of target proteins in the plasma membrane, as shown here for a GTP-bound alpha subunit. The activated target proteins then relay the signal to other components in the signaling cascade.
All right, so I'm gonna pull up another one, that we'll look at again tomorrow, but it will show you a little bit more of, this is showing our regulatory proteins that I'm not, worried about.
This animation depicts the surface view of a target cell and its interaction with a peptide hormone as a model to demonstrate G protein-coupled signal transduction in cells. Peptide hormones generally consist of short chains of amino acids. Because peptide hormones are often quite short, they lack a rigid three-dimensional conformation.
We're gonna have the
and are
surface of the
flexible.
cell, not inside it.
Peptide hormones are usually water-soluble and too large to pass through the lipid plasma membrane of their target cells. Therefore, peptide hormones interact with a cell surface receptor that has an exposed extracellular hormonal binding site. The peptide hormone fits into the receptor like a key fits into a lock, and the hormonal message is conveyed to the receptor. The activated receptor then undergoes a conformational change that transfers the hormonal message into the cell. Once inside the cell, the message is converted or transduced into unique chemicals referred to as second messengers. The intracellular second messengers control the cellular biochemistry and determine how the cell should respond to the hormone. In this example, the hormone stimulates the target cell to open the membrane cation channel. When the channel opens, cations enter the cell and cause a localized depolarization that spreads over an area of the cell surface. In this model, the localized depolarization of the cell membrane represents the target cell response to the hormonal message. Finally, we fade into a view of the internal organization of the target cell that shows the peptide hormone receptor and its chemicals for converting the hormone message into the intracellular second messengers. This scene will show the details of how a target cell converts an external hormonal message into internal second messengers that produce a cellular response. The hormone receptor is a protein that has 7 hydrophobic transmembrane spanning regions that project through the cell's plasma membrane. The receptor is associated with a G protein that is comprised of alpha, beta, and gamma subunits. The peptide hormone assumes its active configuration, fits onto the receptor binding site, and conveys its message. The message results in a conformational change in the receptor that is transferred mechanically to the G protein. Mechanical activation of the G protein results in the alpha subunit replacing guanosine diphosphate with guanosine triphosphate. The activated alpha subunit separates from the beta and gamma subunits and migrates within the plasma membrane to an inactive phospholipase C enzyme. Energy captured from the hydrolysis of the terminal phosphate of guanosine triphosphate is transferred through the alpha subunit to activate the phospholipase C. The process of activating the phospholipase C converts the guanosine triphosphate back to guanosine diphosphate, and the alpha subunit rejoins the beta and gamma subunits to reform the inactive G protein. Activated phospholipase C degrades phospholipids that make up the plasma membrane to produce 2 second messengers, inositol 1,4,5 tris phosphate and diacylglycerol.
We're covering this in lecture tomorrow, so don't.
The inositol
Don't worry about remembering
1,4,5
those right now.
trisphosphate is released from the membrane and binds to a receptor on the endoplasmic reticulum.
Second messenger,
This
moving through the
intracellular signal
cytoplasm.
results in the release of calcium stored within the endoplasmic reticulum. The released calcium plus the diacylglycerol from the membrane activate an inactive protein kinase enzyme in the plasma membrane. The previous scene demonstrated how second messengers transfer the primary message from a hormone to activate an enzyme that will produce a physiological response in the target cell. Activated activated protein kinase from the previous scene facilitates the transfer of a high-energy phosphate group from adenosine triphosphate to an inactive cation channel protein. Phosphorylation of the cation channel protein activates the channel protein and causes opening of the cation channel. Opening the cation channel allows extracellular cations, such as sodium, to enter the cell. The diffusion of sodium into the cell results in a localized depolarization of the cell membrane in the region of the channel. The depolarization spreads across an area of the cell surface. Since this is only a hypothetical model to explain G protein signaling, the depolarization is presumed to produce an important physiological response within the target cell or to signal other cells. G protein coupling.
All right, so I went through that a little bit more, to kind of give you the idea of what we're gonna come back to tomorrow, and it's those downstream effects. It's what's happening in the cell. It's other molecules that are activated that are the second messenger. Now notice the result, that ion channel that was opening for sodium, had the same effect as our ionotropic channel that opened for sodium, without going through all of the rest of these steps. So why would we even want to do that? Ionotropic channels are very quick opening and quick closing, and limited to the number of ion receptors that are actually on the surface of the cell. This has a magnitude or an amplified effect. One receptor binding one hormone can activate multiple the dialkylglycerol or the inositol triphosphate and release lots of ions, opening lots of channels. So it does take longer, but it has a much more amplified, broader effect that can occur inside the cell. And can also give further steps for control. The more steps you are, the more places control can occur. So when you come back tomorrow, I'll briefly review the